I'm sure that you are familiar with NABAR, which is a Nordic forum of Bangladeshi academicians and researchers. Although this is uh, considering a group for only for researcher and academician, but we have an opportunity, and we always invite students also, those who are doing PhD, even master student, because we have that opportunity to link with our group. So, and we are organizing time to time different seminars and workshops. However, for the students, considering our program, this is the first program we are introducing today. In addition, we had some programs with Global Life initiatives in collaboration with Global Life Initiative with Karolinska Institution. So I will not take too much time today. I'll just introduce our today's guest, or he is also our member, Mr. Abu Bakr Siddiq, who is a bioinformatician and working at SLU, Swedish Agriculture University. And I have to thank him for agreeing to talk about this very important software that which we need as a researcher or academician in our daily life, I would say. I think Abu Bakr, please introduce yourself and start. You can start your session. But before
So welcome everybody again. Our today's topic is Introduction to R, and we will highlight a bit on data manipulation, tidying, and data visualization. Maybe you need a little introduction from my side, who I am. Here is my little intro. I am mainly a biologist, but over the time I became bioinformatician by training. My core focus area is molecular ecology, especially metaomics. Within metaomics, I am expert in metagenomics. I also analyze all kind of data within metaomics, like metatranscriptomics, transcriptomics, sometimes metabolomics. My PhD and postdoc was on mostly fungal ecology and eDNA-based analysis. I do teaching sometime within the SLU as well as outside of SLU in Bangladeshi diaspora, as well as for Bangladeshi students from my university or around the universities there. Especially the courses are on NGS data analysis or bioinformatics and R introduction, R foundation. I also teach sometimes basic academic writing. I also help students by giving talk on higher study abroad preparation and so on. I lived in so many places. I started with Sweden after Bangladesh, then went to Germany for PhD, came to Czech Republic, then came back to Sweden again for second postdoc, and since then I'm here. So I have a little bit experience on data analysis. So that leads to this slide where I see this slide as a foundation of today's talk. And what we see, or what I saw last 12 years or 15 years, what we do actually in university or in industry scale. As a bachelor student, as a master student, as a PhD, postdoc researcher, or professor, we generally do data generation. We do data curation or managing data, tidying data, cleaning the data. Then we go to into more data manipulation or transformation. So if I say in the very left, there is environmental data, either you collect from the air or from the mountain or from the river, or maybe it is not environmental data, it is a lab generated data, either molecular biology lab or in the hospital or somewhere else. So data come in so many formats. And all of the data we need to analyze either by a software like R or software like Python or software like Stata and so on, so on. So we basically import our data into that programming language, and then we start organizing our data, which is called tidy. And after that, we start doing transformation. Then we do some calculation, we do some rigorous analysis, some statistics, some modeling, and we also visualize our data time to time to see if everything looks amazing, or the thing I wanted to see, the problem I wanted to see, or the solution I wanted to give. Did I see that? Once we see that, we start to communicate either these results to our professor, to our fellow colleagues, or to a conference, or we publish as a report, as an article, and so on, so on. So this is what we do actually. So as a academic person, whether one is bachelor student or master student or PhD, if he or she can make him very good in that, he could carry a very good life in university as well as in the industry. So today's session is all about, I will talk about R, what is R, why we need to learn R. Then I will give a little R Studio tour, which is a dashboard-like software who deals with R. And then we will learn how we, as a beginner, import a data to a R Studio. And then we try to play with a little bit with the data, and then we try to see if we can discover anything within our data, and then we would like to visualize that data, and so on. So at the end of the session, you will go home with understanding of how to navigate RStudio, how to upload a data, how to tidy, clean your data, and how to wrangle, transform, or manipulate your data. And finally, how to plot, how to make a graph, which you can save and publish later on. So start with the first part, why R. So before that, we need to know what is R basically. R is a programming language, as Dr. Mushfiq said already. It's like, it's originated from S language and was built by the New Zealandan scientists who were statistician, and then it became famous after a while. And it's very famous because it's free and open source. You do not need to pay anything, and you just need to install it and start using it. It is very famous for statistical computing power. We can do all sorts of statistical analysis with R, even when we need a very high computing power with even HPC with the server. We can use R for that as well. So it's also a powerful computational tool as well. It's feature rich. It has almost 24 packages only in Crank R database. Also, the presentation you see today, my presentation was built in R. And R can produce reports, can produce interactive reports, reproducible reports that you can readily show to your supervisors or to the meeting or anywhere. And those features is called Quarto and R Markdown, which is a little bit part of that advanced R. And it's produced very good publication quality graphics, and that is why it is also so famous. And overall, if one learn this R, he can transfer these skills. It's called transferable skills. You can use your skill to anywhere, basically, where there is a need for data analysis. So basically, my position is bioinformatician is also important because of the capacity of data analysis. But R is not a statistician, so you need to validate and verify all of your results yourself. And also, it is not the very best programming language. We know there are many other programming language which is superior than R. The developers, the programmers, they use different programming language like Python and so on. But in academia, R is more elegant, more convenient, and we are very used to with R. And also, it is not most efficient programming solution. But yet, if we compare R with other available tools like Python, Stata, SPSS, and Excel, we can see that R is almost a complete package in terms of free, easy to learn, reproducibility for bioinformatics and machine learning. But there is a little steep learning curve for R. So the organizer or the management of the R group, they produce some tools like RStudio, which is called positron. They try to make it, try to merge Python and R and make positron so that one interested in Python can also learn Python, execute Python in R without leaving R and RStudio. So it's new in the market. You can try it at your spare time. But overall, we see that R is very good in terms of fulfilling the necessity of the current data analysis or data mining. Many people confuse with R and RStudio. They are completely two different things, but one is integrated to another. R is independent tool, which is we call like engine, and RStudio is like a dashboard within our car, where you have engine as inbuilt, but you can also control so many other things from your wheel, from like AC to speed to the temperature. You want to listen something, you want to see something, so on. So everything you can look and control from a dashboard. So if I see it in computer, how it look like, in the left you see R. You can download this tool by this link. I will share this entire lecture afterwards. If you drop in your email in the chat afterwards, near ending of the session, then we will try to send you the lecture. So no worries, you do not need to install R right now. You do not need to install RStudio right now. Just listen, try to navigate what I say and so on. So R is independent. It's like a console. It's like a terminal. You can work with the basic commands and packages, which is inbuilt. In RStudio, there are 4 different windows, and you see they are named. One is called source. I will give a tour a little bit, maybe 1 or 2 minutes after. So I will not discuss it detail now, but later. So 4 window, one is script window, one is console window, which is basically R in the left bottom. And in the right is environment, where you see what you have created, what data you are analyzing, what objects, plots you made. And then in the output, you see plots, you see packages, you see tutorials, even you can see this presentation in this right bottom corner. R cannot run so many things. If you try to run so many commands, you may need some packages. So the packages which are not inbuilt in R, you may need to install those packages. So it's like installing, let's say you have a package called Light, you install it in your socket, and then you call or attach this library every time you want with the command called library. So here library is a command. Every command after the initials, you will see a first bracket. So this is basically a function, and this is we also call command. So you can just turn it on off based on your need, but you need to install only once. One of the mother package called Tidyverse has also so many individuals little projects which are very good, developed by Hardly Pickman to help us who are academician or very enthusiastic to learn R, so that we can learn R at home uh, without installing or installing so many libraries. So Tidyverse will help us to install once. You need to use this command install.package tidyverse, and then every time you call it or attach it with library tidyverse. And it has so many other packages such as ggplot2 is for visualization, readr is for uh, installing or uploading your data to the RStudio. String R is for changing text. TidyR is for changing your data orientation or managing your data, cleaning your data. Deepler is for more data manipulation and analysis. So overall, again, if we like to see the whole game, to analyze our whole game from data collection to data visualization, we also can use all of these tools like as in Tidyverse. So Tidyverse can do all of your job mostly in data analysis. So we are planning after this pre-session a workshop to help you with a more detailed data analysis and data visualization scope, which is not possible by today's session. So today I like to cover everything briefly, but if you need, you can sign for this workshop later. I will come to that workshop at the very end of this session. So let's go to our next part, which is RStudio 2. So RStudio is look like this. It's already opened. So if you install it already, you can just call it RStudio and you see it and you click there, you will see something like that. Initially, you may see 3 windows. This window in the very first occasion may be absent, but other 3 you will see. So in the left is for a script. So if I need to write all my codes in a notebook, I just go to the file and then open a R script. If someone say I would like to produce a reproducible document that I can show to my professor, he can open a quarter document, but one may need a little education how to handle quarter documents and so on. And you can quarter presentation, you can do markdown, so on. So many other steps are available. Also, you can, from here, you can do so many other things, such as, I need to click, such as you may open a project, you may import your data set, which we'll see a little bit later. You can either from an Excel or from a notebook or CSV file, you can import. So in the manual way, you can use RStudio as well. If you want to see where you are working in your path or in the file system in your computer, you can say go to the set the working directory where is my this resource file located. Also, you can have so many other things. You can change color. You see this is black color. You can change it to white or any other color you wish from this global option. In the right side, anything you run, let's say I open a script called untitled R script, which I like to just create for this session. And if you have any code already in your mind, you can copy and paste them, or you can start writing. Let's say what I will show in RStudio, I just have those little lines here. I copy them in the new script. And then I will keep working on that. And you see, when I write a script, I put a hash in the very beginning. So anything you would like to have as a comment for yourself or for someone you would like to share this script later on, you just add a hash after before in the beginning of the sentence. So it became paralyzed. This line will not be activate anymore. So if I run anything here, it will be executed here in the console. Let's say, and this console is also called R, the separate R I was talking about in the very beginning. So anything you use here, let's say I would like to calculate 2 into 2. And to execute this line, I have two options. Either I click here in the run button while I am in that line, or I can press Control Enter in my keyboard, and then this will be executed here in the console 2 plus 2, and it's generated that it is 2 plus 2 is 4. And anything I like to generate as an object, let's say 2 into 2, I would like to assign this into an object which I like to save in R. Let's say I turn 2 into 2 in the result into A, then a sign I used, which is called assignment sign, which is basically less than and a minus. If you put this together, then we call it assignment. So anything after will be saved to this little object. So if I execute this same way, Control Enter, or by just running this, you see something happen in the right screen. Is that 2 into 2, the result is saved as A. And it say the type of this object is numeric because the value of it is 4. And the size you can also say 56 bytes. It's R who actually save it for you. If you make any data, we can also call it vector. Let's say I have some name I like to create. And then another function we use, which is called collect, which is basically C, first bracket and double bracket. And then you just randomly say some name. Let's say Korim. I like Korim. So R cannot allow you to write Korim as a value because it is text. Then you need to provide a which is called probably a quotation mark. You need to put quotation mark before and after Korim or after the text. And then you like to have another name, maybe Rohim. And then you again put quotation mark after and before. And then if you enter, Control Enter or run, you will see another object is made, which is called an object name, and which is type is called character. So any text, any string you use, R will treat it as a character.
I have one question here.
Yes, please.
The insert space between Korean and Rohim, how significant it is?
It is not that significant. You cannot have it as well.
Okay.
So yeah, with or without is fine.
Okay. Thank you.
And then you can have so many things. Let's say I create another object called age, and then I say this 2-person age is one is, let's say, 30 Korim. And now, because it is a number, so I do not need to put quotation mark. Number R can recognize easily. So 30 and Rohim age is, let's say, 40. So I just execute this again, either click here or just Control Enter. And then you see age is created, which is 30, 40. It's also saying which position in the third bracket. So R has its own style of remembering where is who. So 30 is in the first position, 40 in the first position. If you have so many names and age, you will have so many numbers and so many things. And then if you say, I have something else, let's say, let's say your number or your mark, someone has started, give the exam, have marks. One get, let's say, 66.6. Another one get, let's say, 55 point something. And then I can save it as well. And then it also became numeric, but it's in a fraction, or in Bangla we call doshomik, so doshomik number. So if someone says, I like to make a data frame, like we have data, Excel sheet, and so on, how can I make it? So let's say make a data, test data, with these three things which already we have created. So we say, anything you make data frame, we need a function called as data frame. And then, of course, it is a function, so you need to keep the function plus first bracket and second bracket. And then you put name, the thing you created. And of course, you have more than one, so you need comma. Oops. But it will not work because I need to use another function within this as data frame, which is called collect. So that means as a data frame within this function, I need to have a collect function as well. And then you see test data was created. So in this window, you can always control, like as car, what is created. You can click here to see it. So if I click in this little table, it will open a new data. But it is not a data frame, it became a list. So to change it, we may change a little bit of the script to make it a data frame, which I have maybe here to see. Yes, we can either use as a data frame command, and then we need to have those as table. Let's say if table is working, table is another function of the same package called tidyverse. And then we may see these data sets, but still it is one variable. So if we like to create the presentation way, we need to have these three lines together. Instead of doing this, we just have it here. And then I need to put a comma after every line so that I do not. And then you will see where this bracket is closing basically. So if you are in this bracket, you know where this bracket is extended to. So now we see this 2 observation and 3 variables. That means 2 rows and 3 columns. Let's open it again, and then you will see. And it is basically giving a little column names here. We can manipulate this column name as well. So we can rename those names. But it is another part. Today, if I address that, then I will lose other things to address today. And then factors. We also sometimes factorize some specific vectors like this one. Because if we do not factorize, R cannot sometimes do proper statistics in some cases. So this is also, I kept it for the future so that we can learn it in another session, how to factorize, but we do not need for today's session. If someone say, I would like to import a real data, how can I do that? You can do it in two ways. One is manual. So either from the file system, import data set, or you can write some codes here, something. So I will show you first the manual way. You can also do this from here. Here is the import data set option for you right in front of you. So you do not need to find it here. Let me click here and it says you want to from text or with reader or Bezier or from Excel. Let's say from the CSV file I would like to upload. So I click it, you saw a new window pop up, which is actually saying me, please navigate to your computer where the file is. So I click here and I say it is today's workshop and it is in the data folder. I just click data folder and then there is a name called field data, which is a CSV Excel comma separated file. So I just click it, just select and I open it. You can double click as well. So now it will prepare a preview for you. You see a preview is refreshing and it's uploaded your preview and how it will look like when you upload it. You similarly see which package you need and which exact command you need. So let me copy this command for you, but I will do manual uploading. So I just click import. Some people say, no, my first line is also data. So then there are some options you can click, make it, make the first line as well as data. So it's up to you whether you define your column ID or not. So for us, it is column ID, column names. So that's why first row as names. And I just import. And whatever I did, it basically ran it here. You just saw, and then it click for me to view the data, which is field data. And you see it is a penguin data. You have penguin ID. There was a data called penguin data. Some scientists went to Antarctica and then collected penguins from, they did the Adamshumari we call, or in Bangla, and we call, let's say, inventory. They have so many islands in the Antarctica, and then they try to take the data, which species it is, and which island it is, what is their beak length, or we call throat length, and what is their body mass, how much kilo or how much gram they are. So this is the manual way. Now I will show you the command way. You saw I copy the command already. I just paste it here, Control V. You name it, whatever you upload, you will name it, let's say, field data. And then you say, what is the command to upload it? And the command here is read CSV. If you are there, it will give you some hints what you may input. So I say it's in the data folder from the current folder. And then the name of the file is this. As it is text, you saw I had to put the quotation mark afterwards. So if I execute this, this will also do the same thing, and so on. So this is what RStudio at the moment. So if I ask you, what you see here. Basically, you see two numerical variables like thing and 2 or 3 columns, which is like text. So let me stop here and then go back to our presentation. Before that, I didn't address here, whatever you see here. You can install any package from this command in this window. You just click install and then a window will pop up. Any package, maybe your field is using some package for regression or some time series analysis. You type that package. You saw in this repository, R has 24,000 packages. So if you let's say you want to use install tidyverse, you just type it and you see tidyverse is came up. And if you click install, it will install it for you manually. You have plots window. Whenever you plot something, it will appear here. Presentation, my current presentation is popping up here. So report will also pop up here. So far, this is the basic. I like to say, if you have any question, you can ask me if you want. Otherwise, I will stop this and then go to presentation again.
So,
Let me.
anyone has any? Do you have any questions from anyone from the audience? Probably. If you have any question, you can also write in chat box, so we can discuss later also. But please feel free to discuss or to share your experience or your challenges with us. And last up, I'm just reminding you, please share your name, designation, and email address. I received most of from most of you, but those who did not share yet, please share your name, designation, and email address. Thank you very much.
Great. We saw data import, how to import data. So I skipped that part as well. So now we have data in RStudio. Now what? Now I'd like to analyze the data. I would like to find out meaningful results from my data, meaningful equation, meaningful interpretation, or some problems or some solution. And that is 60 to 80% of the data analysis as for a data scientist. So whatever you saw in the university we do, 60 to 80% we do this. And what does it mean by tidy? Tidy means in Bangla gochano, sajano gochano, or it also means cleaning. That means you remove your unwanted data or you make some new data if there is something missing or so on. So tidy has 3 rules. One of the rules is each variable, that means the characteristics or the numbers you want to study, each variable should have a column, such as here you see country. So all the country names should be in this column. And there in the next one you see year. So all the year values should be in this column, and cases, population, and so on. So every variable should have a column. And then you have rows. We call it observation. So every observation should have a row. And the third category is every cell or every position should have a value. So one cell should have one value. Once you have this data ready, your data is already tidy. Your data is already managed, already organized. You have collected a very nice piece of Excel sheet you have made very nicely. So you do not need so much thing further. So in this part, tidyr package, which is a part of tidyverse, is actually reshape the data, help you to organize. And deep layer, they basically clean and transform your values if you need to. So they both work together. Sometimes tidyr comes first. Sometimes deep layer or codes or packages comes first. So let's see. We just saw the data in our RStudio. Now if I call the data, and the call means print, I say R, please print my entire table. And then I just name the object I created in RStudio. So this window you see, this is basically a script window you saw in RStudio, the top left corner. So anything from now on you see as a box, you will understand this is basically script. This is a command where I execute my command. And anything you see afterwards, it is basically console, the left bottom box. So I call it, I print it, I just name it and then run this command, and then it printed entire results. So is this data managed, gochano, sajano, or is it a messy data? This 9 observation data is actually messy data because we have some problem in this 4th line. If you see critically, you will see these 3 problems. So the first problem is we have many values in one cell. We just heard that one cell should have only one number, one values, either it is a text or a numbers. So we need to create another 2 lines to take these 2, right? So we need to split the cell. This we will do today. And also we see that some columns are mixed up, text and with numbers. It should have been a number numeric type or entire column, but it is a character now. That means are turned each and every number you see here into a text inside, internally. We do not see it, but we can see it from here because this is basically printed by R, this line, which is not part of our data, but you can see it is character. That means it is not numeric because of these 2 boxes has too many values and the space. So a space is like text. That's why it turned into. So we need to make this numeric. So this is the second job, to make it tidy. And then some categories, third one, that means some names R is case sensitive. Some names are capital, start with capital letter. Some values start with small letter. You see here as well. Some is completely capital letters. So we need to change this because R treated this name and this name is 2 different names, even though in meaning they are same, basically same island or same species. So we need to adjust those. So I'll quickly go. You may feel it a little bit difficult, but do not worry. We will share this lecture later on. If you miss anything, you can go through and retake, no problem. So first we'll do what? We will do, we should split the rows. Every row should have only one observation. So we will split. To split, there is a command from tidyr called separate rows. So this command will work on this data to make it together, do not have anything in between. We use a special character which is called pipe, though it is, this one is look like pipe, but this one is not. But it is entirely called pipe. What pipe does, you put like as our basin, like as our toilet, in the pipeline, something goes in, it goes all the way to where it's supposed to go. So we connect every command with a pipe. So it doesn't go anywhere else. It just keep working, keep doing the job I wanted R to do. And it's like a chain steps. It's a readable workflow, basically. So when I say fill data, it's basically print this entire data. And then I say, please separate rows. This is a command. Separate these 2 rows, this one, and separate these 2 columns where we have a space. I said separation was with a space. So wherever you see a space, please split there. And it did. It created 2 extra lines for us. So think about it. If you have thousands and thousand rows, if you have a file which is even cannot open by a computer, it need to be opened by a computer server. Then you just simply use R to split those. If it is a small, you can do it in Excel, copy pasting and changing. But if it is too much, you cannot. So that helps us. So we split it, we make it seems more tidy now, but still we have problem with the name and with the cases. So next, R is case sensitive, so we will change those. So we'll make this P capital, this small, and this small one capital, and this change first one into capital and the rest into small letters. So how can we do that? There is another command called mutate, which came from Dipler. And there is another command came from string R, where string can be changed. We say, please take the string, that means take the text and change it to title. So title, we know in the title we have capital letter in the beginning and then all the letters are smalls. So we say, please, we have problem in this 3 column, penguin ID, species and island. Only work on those 3 columns. And then we mention what are those columns. And we did that. What it does, mutate, it basically overwrite these names. It take the names and overwrite and then paste the current name on it. So then we can say, okay, our data looks now tidy. But we have still a problem because it is not numeric. If we plot something or calculate mean or average, this will not work because it is still character. So we need to change those column character to numbers. How we do that? We do another mutate. We absolutely we use pipe because we are not done yet. So we use pipe in the just at the end of last command, and then we keep continue these 2 lines. We say first one should be numeric because it is dashmic or decimal thing, and then we say this is like absolute number, so it should be integer. So R rename this number as integer. So now you see R make it numeric, which R itself know it as a double. So every numeric, whether it is dashmic, decimal, or it is absolute value, it treated it as a double. And then I say make this body mass column integer, and it did that. So now we can say every variable has its own column, every observation has its own row, and value has each cell. So we can call it tidy. So recap what we did so far. We have attached the library. I have done it before today's session, so I didn't run it, but you need to run it if you already installed tidyverse package. And then I imported my data and I just did 3. Sorry, it just took me backward when I click unintentionally. So it's basically the third one I did is just tidying my data or cleaning the data, and it became this. So just 14 lines of codes just equip me to install, to upload my data and clean the data. For field to field, it varies. For some people, it takes many lines. Yes.
Mr. There is a question from Asif Mahmood.
Yes, please. What is it?
আসসালামু আলাইকুম, আমি কি বাংলায় কোশ্চেনটা করতে পারি?
Yes.
Sure.
এখানে হচ্ছে টাইডি অর 3 ডাটা অংশে আমরা যে মিউটেটটা করছি স্পিসিস, পেঙ্গুইন এবং আইল্যান্ডের আলাদা আলাদা করে লিখছি, যে এইটা কি আমরা একটা কোডের মধ্যে লিখতে পারি কিনা? মানে স্ট্রিঙ্গার টু টাইটেল, এটা স্পিসিস, পেঙ্গুইন, এটা আইল্যান্ড, এই 3টা বা আরো বেশি যখন থাকবে কলাম, তখন কি একটা লাইনের মধ্যে লেখা যাবে কিনা, নাকি প্রত্যেকটা আলাদা আলাদা করে দিতে হবে?
Very good question. I haven't checked it. Maybe it can be done if the programmers who actually build this mutate, he kept a option for us. I hope there is option, but if not, this is the basic way. So you may try. This is homework. So try it if you can use collect option, see, and then keep everything within this loop. But it may not. So from my understanding, but just try Google or just ask AI. Maybe there is a solution for that to make it more little. Thank you for the
অনেক
question.
ধন্যবাদ। আরো দুইটা কোয়েশ্চেন আছে আমার।
Yes, yes.
আরেকটা হচ্ছে এই যে নট অ্যাপ্লিকেবল যেই ডেটাগুলো আছে এগুলা কি সিস্টেম অটোমেটিকলি করে অ্যাসাইন করে, নাকি হচ্ছে আমাদের কোনো কিছু করা লাগে? আর
Yes.
আরেকটা হচ্ছে ওই যে ফিল্ড ডাটার পরে যেই চিহ্নটার কথা বললেন, এটা আমরা কয়েকবার ব্যবহার করতেছি কোডের মধ্যে, এইটার ব্যাপারে আরেকবার একটু যদি রিক্যাপ করতেন। অনেক ধন্যবাদ।
ওকে, প্রথম প্রশ্ন ছিল যে এনএ ভ্যালিউ যখন মিসিং থাকে, কোনো ভ্যালিউ না থাকে, তখন আমরা যখন আপলোড করি আর ট্রাই টু গিভ ইট অ্যাজ এ এনএ itself, and it will tell you that I have replaced all the empty blank space with NA. Sometimes it doesn't. So it's up to the command that actually importing. So if let's say CSV, if it is a read CSV command, it may assign NA value for yourself without needing anything. So all the blank space is converted into NA. For some other reasons, let's say read XLS, it may not. So you have to see. And I intentionally kept it away because otherwise this session will be too long. This is also part of data curation, data cleaning. So there are so many other steps for data cleaning. One of is this NA, removing NAs, either removing all NAs or removing specific NAs. Let's say here I just want to delete, let's say line number 10, this line, because it has a species which is NA. So that means it is not relevant for my answer because I do not know what is the species. So I just delete it. Some people say, no, I am doing statistics. I cannot allow any NA. So then you have a command, say, drop NA. We will see that command in a moment, maybe afterwards. And the second answer is, what was that? Can you hint a bit?
আপনার ফিল্ড ডাটার পরে যে চিহ্নটা আমরা ব্যবহার
Yes,
করছি ওইটা
it is
কোডের
called
মধ্যে।
pipe. It is actually an advanced R, but I bring it here so that it looks nice. Otherwise, so many steps you will lose. I would like to have a chronological way to explanation. When you use, things go forward. That means field data will go to next step. Next step is separating row. And then this is done, it will go to next step called mutate, and then so on. But it will never go opposite. So you cannot run separate row first and then go to field data. So it is like a pipe. It's scrolling, let's say, towards the end, like our rains. When we have rains in the in the roof, and then it goes with so many pipes to the downstreams like this. Then we have a little bit did tidying. Now we like to discover something. Let's say if we have any penguin which is bigger in size, let's say 5 kg plus, how can I only have 5 kg plus penguins in my data, not anything else? I like to do that. So this is called data transformation or data wrangling. We do this with the deepler. Deepler has so many other commands like filter. Filter command basically pick only rows that has a specific condition you mentioned. Here I said 5 kg. So we can use filter to have rows only which has 5 kg body mass. Select, we can just pick couple of columns, not all the columns. We have thousands and thousands of columns in some data. We cannot see them at once. So then we use select to pick some columns. We use mutate, we already saw that, to create modify columns. And again, pipe is just reads left to right like a sentence. So we connect everything with the pipe. So let's see, how can we have only 5 kg penguins? We have this data, and you saw that command already. So this is already you know. I just give you a hint. Here we have 5 kg, so that means 5,000 grams. So we have three rows. We have 5 kg, 5 rows, 4 rows basically. So I like to have only those. How can I do that? We use an extra command called filter. Within this filter command, in the bracket, we say which column it is. I say it is body mass G, and the condition is greater than 5,000. If number is greater than 5,000, then it will filter. That means keep. It will keep rows which has greater than 5 kg. And you exactly saw this. So everything in the beginning was same. You saw that. So I just make it not highlighted, but only the new code highlighted. And I put a comment on it so that I know what it is, what the next step does. So it just filter out rest, but only the 5 kg thing. And then you like to say, okay, I like to have a kg unit, not the gram, because it's not looking nice. So we need a new column. So how can we create a new column from an existing column? This is one of the important aspects of data analysis. So here we add another line with pipe again. We say divide the body mass gram with 1000. We know 1000 gram equal to 1 kilo. So we have a new column is this, but the function was here muted. So this is the way we go downstream and we make new column, new value. Some people do log transformation. Some people do CLR or something, advanced stats. We can do that. There are many functions. We will see those in the future sessions if we are able to do. Recap. So again, package, we did data uploading, tidying, and did a little bit data wrangling. Now, the last step, which is data visualization. We are happy what we saw. Now we like to visualize what we saw, or what we have. And that is 10 to 20% of the data analysis. So a data analyst do 10 to 20% of his life on data visualization. How we do it, why it is important. It is essential because it is a part of data analysis, because there are so many data which has similar mean, standard deviation, correlation, but still when you plot, it is totally different. In the left, you see Uncom squatted data, which has similar mean, standard deviation, but completely looks different. In the right, you see 13 data points, which is Datasaurus data. They have similar mean, standard deviation, correlation, but when you plot, they are looking completely different. Even one is looking like a dinosaur. So data, looking into Excel sheet, looking into data is not enough. We need to visualize what they tell. Do they tell something else? How we do it? How do we visualize? One is base graphics, which R has inbuilt. And another one is called ggplot2, which is a package, an advanced package made by Henry Bigman. And because of this package, R also gets so much popular. So first see some base graphs. So base graphs is very easy. You just ask the plot function, and then you define which data it is, and then you say which column it is with a $sign, and then it plots you. So you remember, I just rename our new transform data into field data 2, where we have 4 rows we saw. We just did a scatter plot with this command. We did a box plot, which may be statistically for some people important because we look at significance level by this plot. Some people say, I like to bar plot, then you can bar plot. Some people say, I need to see distribution, then you ask for histogram. Now, if we compare with ggplot with a more bigger data, let's say we like to use the main original penguins data where we have almost more than 400 rows of data. And from that data, we try to call a base graphics with histogram. And then for ggplot, this is actually the command look like. Initially, it looks ggplot needs more commands than the base commands. But once you try to publish something, you need more commands for basic rplot, which is too messy. You need 6 lines to plot this left plot. And in ggplot, you just needed a little extra thing to plot in the right, where you have a free legend, you have free colors as well, though you have we have mentioned colors here, but it automatically assigned the color for you. So once you make advanced plot, ggplot is more better. So why ggplot? It's consistent code. You saw I can increase the code a little bit by little, and then it gets more nicer without leaving us behind what has happened. It's flexible. You can remove some part of it, but only thing you remember, you add new command with a plus at the end of the previous line. So previous line, we added a column. We see that in a minute. It's create automatic legends. It's say plot as an object, which you cannot do with base plot. Numerous extension, it is complete a graphic solution right now. And what is the grammar of it? It's basically take your data first, and then you mentioned where it goes, who goes where, which column goes to X, which column goes to Y, and then which kind of plot you want. You want a scatter plot, bar plot, or line plot or something. And then some people say, I like to divide my plot 2 into many plot. Then we use facets, we use stats inbuilt. We can have error bars, standard deviation, so on. We want to have coordinates. Some people like to use as map or something. And we like to have a nice background color, theme, and so on that we can control with grammar of graphics. How we build a graph in ggplot completely out of nothing. So from the very beginner, you need a syntax on which you can keep in mind, and then you can use it. So here you see a syntax. First 2 lines are important to plot. The rest are optional. You do not require right now, but you can use those for your scientific purpose, for your data, for your field. But first 2 lines is enough to create a ggplot. So what basically here, it is a ggplot command. We know every command starts with a bracket, and then you provide your data, and then a plus sign. It's like a pipe thing. The philosophy is same, but the thing is different. So then you say, I have data. Now I would like to have aesthetics and geometry. So geom function is geometry. You mention it, whether it is a scattered plot or something, and then you mention what is X, what is Y here. And that's enough to plot. So let's see how to plot. We need a data. So we ask for Palmer penguins data. They collected, some scientists went to Antarctica, collected 3 species data from different islands, and they measure their beaks, their flipper length, and then weight, and so on. So we attach the data with data penguins. So this command is ggplot, and we provided data is penguins because we just saw we attach penguins. And then you saw a canvas. So nothing is plotted, but data is there. So even though it's blank, but data is already there, but it is not showing anything because we haven't provided any further commands. So let's say we like to have something in X here and something in Y. So we add that in a mapping, and it is called aesthetics. We say in the X, please send the column bill length millimeter, and in the Y, please send body mass in grams. And then it provided. So X and Y was marked for me, and it says their range, it is in gram, it is in millimeter. But still, we do not see any plot. What happened? Because we haven't mentioned what kind of plot we want. We want a scatter plot, we want a line plot, we want a bar plot, so on. So let's say we want a scatter plot. And then we provided that with a plus and geom point. So this part will change based on what you want. So if you say line, then you say geom line. If you say bar, then geom bar. If you say geom box plot, then geom box. Now we see a nice graph where we see length, build length versus body mass. And we see some like correlation, something. If something going high, body mass is going high. But you want to know which species they are. They are from same species. You want to have another column as a color. So let's add a color in this aesthetics. So color equal to, now we will say species. Now we add with the comma color species. And then we saw which species it is. And this entire line 2 to 4, we can put in geom point as well. It is another style. So people can play with this. Either you can have as it is within ggplot command, or you can have line 2 to 4 within geom point, how it is look like if you have it here in geom point. So it is look like this. Entire, this comes from here. And then if you look at the syntax now you see it is exactly following our syntax, these lines. So my purpose is served. I wanted to show how a ggplot can be made, a publication plot. But you say, I like to show it to my professor, or show it to a conference, or show it to a meeting. How can I save this plot you just created? Then you entire plot, you either assign it to a name, like you saw I assigned in an object. So here we assigned it entire thing to object P, and then we print P with this. You already know this. Now we would like to save it. Some journals may say, okay, your graph quality should be 200 dpi, and then your graph should not be more than 5 centimeter. That means 50 millimeter long, and wide should not be more than 7 centimeter. So then you mention those, and you save it with the command called ggplot. You name it, which name it should be, and the object you want to save. So here it was P, so I mentioned it is P. If it is something else, you need to write it here. And then it produce and save your graphs in your existing location where you are running this R. Or you can define it as well here in this box. You can also save in a base graphics way. This is a base graphics way command. I do not explain it what it is, but you will see this is a common practice. So I am now running quick so that I can finish my presentation. And in the workshop, if you're interested, we more look into that, different aesthetics, advanced plotting, how to combine plots, how to number the plots. If you want to publish, you sometimes need to have many plots together with serial number A, B, C, D or something. How can we do that? Sometimes you need to have stats. Sometimes you need to have multiple plots, bar plots, point plots together. You need to sometimes have colors. How can we will see those in upcoming sessions. Some people are very interested in statistics. Here is a little command, which is inbuilt command in R called summary. We call penguins, and then as it is statistics, we say, please drop all the NA values. Now you see here how we drop NA values. So all the NA values will be discarded, and then you will execute summary command, which tell you each column summary. So a species column, it says, I have 3 species, Adelie, Sinstap, Gentoo, and how many of them. And then in island, how many island I have and what are the name. If it is numeric, it will calculate the quartile, the median, mean for you. So this is good to have a summary on your data in the very beginning to work further. In upcoming session, we may look into more t-test, how to do chi-square test, how to do linear regression, generalized linear modeling, PCA, principal component analysis, or non-metric multidimensional data visualization. Some people interested in biological QA, which is a part of bioinformatics. So on, how to handle missing values, and so on. So these will be next sessions. So putting it all together, what I have discussed so far. So I showed how to install or attach package, how to import data to your R, how to tidy your data or clean your data. And then I showed how to wrangle, transform data. We wanted to have only 5 kilo penguins. And then we wanted to plot. Even you can directly plot from here. We using this exact 5 column, you directly say, please plot ggplot. Then you do not need to mention data because data is already created for you in the previous command. Will directly come here and will be plotted for you. Great. If you need further help, you do not want to have a workshop, you want to learn yourself, then these books will be good for you. R for Data Science by Hadley Bickman. And these are online available thing. You can read it. You can ask your AI. Now AI is very good to generate your answers, your codes based on your questions, your prompting. Or you have POSIT community. The POSIT is the one who actually manage all the R softwares, all the R development and managing all of it. Stack Overflow, which is almost dead now because of AI, but still Stack Overflow will help you to answer something. You are interested some chit chats you want to attach in front of you in the wall when you are analyzing data, you can do that as well. So takeaway, R and R Studio is very powerful, reproducible data analysis you can do with it. Tidy R Dipler gives you tidy, clean, and wrangle data. ggplot helps you to plot publicable quality plots. The pipe is a step by which you change all steps. And ready to use in the R. And the final quote, the greatest value of a picture is when it forced us to notice what we never expected to see. Now you see a very clean picture where you clearly see these 3 species are very separated in terms of bill depth and bill length. And you see the more bigger penguins, the more weight they gain when they have bill length more and bill depth more. They can eat more. So this kind of a striking thing you come up with when you have a picture or a plot. Thank you. And I acknowledge Slubi, where I work, and Rauk R, it is a 10 days R workshop, advanced R workshop by the Uppsala University. It's they do in every summer in Gotland for 10 days. And thank you all. If you need to know how can you register for the upcoming session, you can scan this code. You can go to this form. It will take you to this form. As I will paste it later on, maybe. Or you can paste your email address in the chat. Then we will send you an email with these forms and details, what is coming in upcoming workshops and what would be the fee and so on. Yeah, thank you. That is all from me.